Wow, what a beautiful day. Nothing like relax. Uh-oh. What are you doing out here, Dr. Romano?
Hi, I'm Dr. Romano, professor of organic chemistry at Romano Scientific and the creator of the Man products and the DAT Destroyer book. I'd like to go over with you something very important and a major component of the eukaryotic cell and in all animal cells, and that's the molecule cholesterol. So let's have a look. And in this video, I'm going to be combining not only biology, biochemistry, but a considerable amount of organic chemistry you need to know. The most abundant steroid in humans and in all animal cells is cholesterol. It's important in membrane components, even in nervous tissue. Very recently, German scientists have found glial cells actually make cholesterol to supply nerve cells for synapse development. Cholesterol is a major player in vitamin D as a precursor to vitamin D. It's a precursor for steroids. It's a guaranteed type of question you'll see on your exams on the MCAT, on the DAT, the OAT. These precursor hormones, which the steroid hormones include estrogen, progesterone, testosterone, cortisol. It's also a precursor for the bile salts, sodium glycolate, sodium taurocholate, and they're involved in emulsifying fats. Cholesterol is supplied by the diet. Think meats or dairy, but most of it is made by the largest internal organ, if you remember, when we did a clip on that, and that was the liver. Now, the structure of cholesterol is no joke. If you looked over here, this molecule is originally made from acetyl coenzyme A. If you remember, the acetyl coenzyme A was the star of the show because that's the molecule that went into the TCA cycle. Now, many steps are involved in forming cholesterol. Now, one thing is a good exercise I did is I starred all the chiral carbons for you. So let's count them together. This is chiral. Notice there's 1, 2, 3, and then there's invisible H. That makes there's 4 different groups or paths you can take. So we have 1, 2, 3, 4, 5, 6, 7, and 8. There's 8 chiral centers, and if you remembered, we used the rule 2 to the n being the number of chiral carbons here. So 2 to the 8 gave me 256 possible theoretical stereoisomers. As crazy as it sounds, nature only used one of them. Now, what I like to do and just show you that we're going to do a very small portion of how we form cholesterol. And there's a molecule, an intermediate called lanasterol. Lanasterol is formed along the way. I want you to just take a look at the complexity down here, where we have this intermediate molecule, an intermediate called squalene intermediate, and as you can see, an enzyme is going to give an H, and it's going to cause what we call an in vivo cascade rearrangement. Now, as you see, watch what's going to happen. Once we protonate this epoxide, all right, now watch what happens. Look at the way the arrows move. I put a little dot here so you can sort of follow it when we get to the next structure. Okay. But this moves here, this moves here, this moves here, and this moves here. And we get a cyclization. If you follow the dots, you would be able to easily connect those dots. And as you can see, you can use this group here. You can see that group is here. Now, once we've cyclized it and we convert it into this, the cascade rearrangement continues. Now, this arrangement is going to be a 1,2 hydride rearrangement. Then we're going to have a 1,2 methyl rearrangement, and then an elimination. As you can see what I've done, you got this nice open tertiary carbocation. This H moves to here, and this H moves in its place. That's the 1,2 rearrangement of a hydrogen. Now look at the 2 methyls. 1 methyl moves here, 1 methyl moves here. So if you can follow those methyl groups, and then finally, this H comes off, and that would finish the elimination. That's going to give us the molecule lanasterol. So you should be able to follow the arrows and understand, for instance, when I moved this to here, that laid down the double bond. The way they got these 2 methyls, this moved here and this moved here, and we came up with that. And then, of course, this hydrogen was from the shift on the tertiary. And many steps beyond that gives you cholesterol. I hope this gives you some appreciation for the complexity. That was a lot of steps, and that was only a little piece of the mechanism. But as you can see, we want to make sure we're ready for the bio. We know, for instance, if I say, where is cholesterol made? You're going to tell me the liver. That's a guaranteed type of question. You're going to want to make sure you have understanding of, like, it's a precursor to the steroid hormones. It's built from acetyl coenzyme A. These are the questions that are important for the DAT. But also, a very common question, at least on MCATs, is they would give you a molecule like this and say, how many chiral centers are there? You should be able to easily tell me there's 8, or a molecule just like that. Or if I pushed it a little further, how many stereoisomers? You would say 2 to the n, 256. And then I would like you to have some appreciation of the cascade, how an arrow movement gives me another compound. Hope that gives you an idea and a good understanding of the complexity of this molecule called cholesterol, which is pretty much limited to the eukaryotic organism, but is found in all animal cells. All right, I hope this helps, and the next video, I'm not even sure what I'll do yet, but I'll think of something really great for you for tomorrow, and this whole week I'll be doing more and more videos. Save your money on videos. I'll do them all for free for you, and there's many PhDs out there, if you hunt on YouTube, that you can find really great videos on biology. All right, good day.
I like your videos, Dr. Romano. Hey, there's boats over there. Want to go on a boat ride, Dr. Romano?
No boating. Time to study. Good day.
Good day to you, sir. Thank you, Dr. Romano. That man never rests.